fine-tuned biological systems are?
In biology, microscopic changes at the atomic level can have amplified effects that we can see, observe, and experience. This is often how disease is caused. For example, a single alteration of a gene in one cell can result as detrimental symptoms. To give you an idea of scale, there's over 37 trillion cells in the human body, and a single cell contains over 100 trillion atoms. So when I talk about a mutation in a gene, I'm talking about switching out a handful of these atoms in one cell that results in the entire disease. On the other hand, we can harness this same phenomenon, seemingly tiny tweaks at the atomic level of biological systems, to improve a protein, a cell, an organ, or an entire organism in really large and beneficial ways. This is synthetic biology, using biological systems as machines and manipulating them for precise purposes. In my Keystone, I used synthetic molecules, so molecules that I made in a lab in order to change a naturally occurring protein. I was investigating the effects of bioincorporating a synthetic amino acid into a model protein, green fluorescent protein. There's a lot of really big words in that title, so I'm hoping in the next few minutes I can give you a sense of what I was doing. Let's start with the basics. I was trying to manipulate the function of a protein by altering its structure. So, what is a protein? Proteins are everywhere, and all biological systems are made up of so many proteins that carry out myriad functions. And so proteins are these really big molecules made up of smaller building blocks or amino acids. The remarkable thing about this is that all of proteins are made up of the same 20 amino acids. So all of the proteins in our cells and in the cells of the trees outside and in the bacteria at the bottom of the ocean are made up of different combinations of the same 20 building blocks. And then these amino acids are strung together like beads on a string in long chains called polypeptide chains. These chains then fold up on themselves in a really specific way to create a three-dimensional structure of what becomes the protein. The structure of the protein is then determines what the function of the protein will be. If you think of a cell as a car and you have all the proteins, which are the parts of the car to make it run smoothly, if you were to change the shape of the wheels to, say, square, your car isn't going to run as well. The same goes for proteins. By changing the structure of the protein, you also affect its function. And in terms of proteins, the structure comes down to the order of these amino acids. So let me give you a real-life example of how the order of these amino acids is so important. Sickle cell anemia is a genetic disease that causes your red blood cells to be sickle or irregularly shaped, and it results in a whole bunch of health complications. Sickle cell anemia is caused by a single mutation in the hemoglobin protein. So hemoglobin is a protein that's made up of over 300 amino acids, and by switching one of these amino acids to another naturally occurring amino acid, it results in the entire phenotype or all of the characteristics of this disease. And this is to give you an idea of how specific the order of these amino acids are. Now, the order of amino acids, or using sickle cell anemia is an example of how detrimental it can be to change the order of these amino acids. But we can also use the specificity of these building blocks as a really fascinating tool to alter proteins and adjust their functions in beneficial ways. Remember that there's only 20 naturally occurring amino acids. So what happens when we use unnatural amino acids, or amino acids that aren't genetically encoded? So our cells don't have the instructions to incorporate them. If we go back to our car analogy, this is like buying aftermarket parts like snow tires to actually increase the function of our car in specific conditions. And this brings me back to my project. So I was investigating some properties of green fluorescent protein, or GFP, which is a naturally occurring protein in a type of jellyfish, and it fluoresces green under UV light. The protein itself, though, is relatively small and sturdy, and so it's often used in research as what's called a reporter protein. So scientists will use this protein to ensure that the system that they are investigating is working, because if the protein is folded correctly, it will glow green under UV light and you can physically see it. If we look at a more molecular model of the protein, On the left, I have an atomic representation of the protein. So this is showing where all of the atoms in the protein are. Essentially what I want to show you here is that GFP is essentially a cylindrical blob. On the right is a more structural model. So remember that amino acids are like beads on a chain. Especially what this green stringy bit is showing you is where the string would be without any of the beads on it. And you can see that it makes a really beautiful symmetrical cylinder shape. And in the middle of this cylinder, which I've shown in blue, is a molecule called a fluorophore, which is what is responsible for the characteristic green glow of GFP. So when the molecule shown in blue gets excited by UV light, it absorbs that energy and releases it in the visible spectrum as green that we can see. The entire purpose of the molecule surrounding the fluorophore is to create the perfect molecular environment so that the fluorophore is happy enough to hold on to that energy and emit it as green. But, as we've talked about, changing small things about the structure of a protein can affect its function. So if we change parts of the green stringy bit of the protein, we may be able to change how much energy the fluorophore can hold onto, thus changing the color that it emits. And this is the core of what I was doing. I tried to change some of the structures of the amino acids in this green stringy bit in order to change how much energy the fluorophore can hold onto, changing the color that GFP glows. If we go down to the amino acid level of what I was doing, I was replacing methionine with a synthetic amino acid. So methionine is shown on the left, and it's a naturally occurring amino acid that shows up in the GFP structure. And I was replacing it with either monofluoroethionine or ethionine. Both of these are synthetic amino acids, and they look really similar to methionine, but they actually contain chemical properties that don't exist in living proteins. Changing an amino acid in a protein isn't that simple, and you can't just build a protein in a lab. So what I did instead is I used bacteria as my machines. I fed the genetic instructions of GFP to E. coli and essentially made GFP factories out of bacteria. So on the left is what GFP looks like, or what E. coli looks like when it's expressing or producing a lot of GFP. And on the right is what bacteria look like under UV light when they can't produce this protein. So now I have a bunch of E. coli that can produce the protein that I'm interested in studying. But I need to switch out the methionine for an unnatural amino acid. Basically, I starve the E. coli of all natural methionine so they don't have access to methionine and feed them a whole bunch of the synthetic amino acid that I'm interested in studying. And then I hope that their cellular machinery can grab onto the synthetic amino acid by mistake in order to incorporate it into the protein. And then I extract all of the protein from the bacteria and compare it to regular GFP. So I don't have any results for the samples that I fed monofluoroethionine to, but this is what occurred when I put ethionine into GFP. So on the left is wild type, or just regular GFP, and on the right are the samples that were starved from methionine and fed ethionine. And you can see a slight shift in fluorescence. So I haven't done any formal analysis of these samples, and this data is very preliminary, but it does support the notion that by changing the structure of the amino acids, we can alter its fluorescence. So what does this mean? Well, as I said, ethionine has chemical characteristics that don't exist in living systems. And if we can put this amino acid into GFP, we may be able to put this amino acid into many other proteins and adjust their functions in unprecedented ways. In that case, we could use GFP as a reporter protein. So if we want to put ethionine into a protein that doesn't glow, we can see if our system is working correctly, if our bacterial machines are really putting the ethionine into the proteins, because our GFP will glow a different color. The applications of synthetic biology is essentially limitless. But I hope what you can take away from this is that changes at a level that are seemingly invisible have effects that we can see with our own eyes. I'd like to thank my mentor, Mark Vaughn, for whom this project wouldn't have been possible, to all my friends and family for the support over the last 22 years, and the Quest Research Scholarship and Creative Works Committee for funding this project.